Welcome to a special feature presentation of The Noisy Code, where we explore the hidden information within our biological systems. Today, we are taking a very deep, comprehensive journey into the next frontier of oncology. For decades, we have studied cancer by its genetics, grinding up tumors to see what genes were active. But what if the secret to understanding cancer isn't just the ingredients, but in the recipe? What if the key is the tumor's architecture? Here to guide us in this journey is Rudranshu Paul, a master's candidate in chemical and molecular biology at the prestigious IIT Kharagpur. His work at the cutting edge of the field that is revolutionizing how we see, understand, and fight cancer. Rudranshu, thank you for joining us for this very extended conversation.
Thank you for having me, Ritika. It's a real pleasure. I'm excited to dive in.
Before we get on to the cutting edge science, I'm curious. Your background involves a shift from traditional wet lab biology to intensely computational work. So, what drew you to this specific frontier, and what was the moment you decided to become, a map maker, so to say, for tumors?
That's a great question because it gets to the heart of why this field is so exciting. I was working in the lab generating what I thought was beautiful data, running gels, doing assays, but I had this nagging feeling that the deepest secrets were locked away in the patterns, not only just the individual data points. I realized that learning to code was like learning a new language through which I could directly speak to the data. It was a revelation. My wet lab experience is invaluable. It gave me an intuition for the biology, for the nuances of the experiment. But the computational skills which I am embedding allows me to take a massive chaotic dataset and find the hidden story within it. It's that duality, the ability to bridge the gap between the two worlds that pulled me within.
A bridge between two worlds. I love that. So let's cross the bridge. Your work is in the field called spatial transcriptomics. And for our listeners, let's begin from that very specific term. So, what does new dimension does adding the spatial context reveal? And why is understanding the architecture of a tumor so critical?
Okay, ma'am. So, this is a central question of the entire field. The difference between traditional and spatial analysis is like the difference between a smoothie and a fruit bowl. If I give you a smoothie made of strawberries, bananas, and spinach, you can put it in a machine and get a perfect list of all the ingredients. That's traditional genomics. We essentially grind up the tumor and get a list of all the genes and cell types present. It's incredibly powerful, but in doing all this, we lose the entire structure. We have no idea how the ingredients were put there or how it were clustered together. But if I give you a fruit bowl, you see not just the ingredients but how they're arranged. You see the strawberries are clustered over there, spinach is tucked in next into the bananas. The arrangement, the architecture is everything. In a tumor, this is critical because the cell's behavior, whether it grows, hides, escapes, is dictated by its neighbors. These interactions are what we call the tumor and microenvironment, and the local environment drives everything from tumor growth to drug resistance to EMTs. Finally, seeing the fruit bowl, we can begin to map the cellular conversations that determine a patient's outcome.
Right. So that's such a very clear analogy. And for years, the big revolution was single cell sequencing, which allowed us to see all the different types of cells in the smoothie, not just the average flavor. So it gave us a census of the tumor's inhabitants. So, how does spatial omics take the leap forward?
Exactly. Single cell was revolutionary. It was like getting a complete census of the city, identifying every inhabitant, the bakers, blankers, police officers. A huge step up from just knowing the city's average resident. But a census, however powerful it may be, doesn't tell you how the city is organized. It doesn't tell you that all the bankers work in a skyscraper downtown, that the police are patrolling a specific neighborhood, or that there's a shady back alley deal happening between two groups. So that's where spatial transcriptomics comes in. It's the difference between that the sensors and a real-time GPS map. We are no longer getting a list of cells. We are seeing their exact coordinates, the XYZ axis. We can now see how the cancer cell behaves one way when it is being suppressed by an immune cell, and a completely different way when it is in a hypoxic environment, that is a low oxygen niche. These neighborhoods actively shape a cell behavior. By combining the what from single cell analysis with the where from specialomics, we can finally start to understand the social networks of the tumor, which is the key to dismantling it.
So it's really about decoding these neighborhoods. So let's talk about a concept you can focus on, like let's say gene expression noise. To many, noise sounds like a random error, something to be filtered out. But you're saying it's a very crucial part of the tumor strategy?
That's a fantastic point because traditionally we treat it as a junk data. We thought of this variability like a static on a radio. Our goal was to tune it out to hear the main broadcast, the average gene expression. But we are now realizing that within this static, there are hidden messages. For a tumor, the noise isn't a flaw, it's a feature. In my field, we distinguish between intrinsic noise that comes from the random mechanics of the transcription itself and the extrinsic noise from the fluctuations in the environment, cell type differences, cell stage differences, etc. Both contribute to tumor survival strategy. It's a way of hedging the bets. By allowing this diversity, the tumor ensures that no matter whatever challenges it faces, like a new powerful therapy, there's always a new group of cells by pure chance that have the right set of tools to survive and regrow. Think of it like trying to open a complex lock with a single key. Our targeted therapies are often like highly specific keys designed to fit only one protein. If every cell in the tumor used the same lock, the therapy would be a cure. But because of gene expression noise and heterogeneity, the tumor doesn't have one lock. It is a diverse toolkit consisting of thousands of different kinds of locks. So our therapy key might open 99% of them, causing the tumor to shrink dramatically. But the 1% of the cells that has a different locks. These are cells that are too noisy enough to have a different internal state. They are free to multiply. They are kind of persistent kind of cells. That is, a new tumor is made of new differently kind of cells that our original key cannot open. My research aims to map this entire toolkit of locks to understand the entire diversity before we even choose the key.
Right. So mapping the toolkit sounds incredibly data intensive. You mentioned a single experiment is like being handed a library of 10,000 books. As a computational biologist, how do you even begin with approaching this data deluge? And what's the first step in turning the wall of raw numbers into tangible and biological insights?
That's a fundamental challenge, and frankly, what makes this field so exhilarating. You're right, the scale is immense. You can't just start reading word by word. The first and most critical step is what we call data process pre processing and quality control. It's unglamorous but essential work of a librarian, organizing the books before anyone can read them. We are computationally checking for errors, filtering out the low quality cells that might be damaged, and normalizing the data before we can make fair comparison. This ensures we are taking a look at the clean, reliable data sets. But the real journey into decoding complexity begins with a concept called dimensionality reduction. We have data in over 20,000 dimensions, one for each gene. The human mind can't comprehend that. So we use algorithms like PCA and more powerfully UMAP to distill this complexity into a more visualizable form. Think of this way: to understand a car, you don't start by measuring the position of every atom. You start with its key features like wheels, engine, chassis. The UMAP does this for cells. It looks at the entire transcriptome profile, all the 20,000 gene expression values, and learns the most important features that define its identity, and then plots each cell on a simple 2D gene expression map. When you do this, something incredible happens. You don't see a random, meaningless cloud of points. You see the complexity spontaneously organize itself into distinct islands and continents. These are our cell types. All the T cells cluster on one island, the cancer cells from other, the fibroblasts from third. So different cell types form different clusters. The first time I ran this analysis, it was breathtaking. It's a glimpse of order emerging from the chaos, giving us a complete census of the inhabitants of our tumor city.
Right. So you create this beautiful atlas of cell types. But as you said, the power is knowing where these cells live. And how do you bridge the gap between, the abstract 2D map of islands and the actual physical tumor? So how do you put the spatial back into spatial transcriptomics?
This is the step that truly defines the field and where the most profound insights come from. We take that UMAC generated cell type identity for every single cell and project it into the high resolution image of the slice, original tissue slice. It's like taking the black and white satellite image of a city that we talked about and color coding every single building based on our consensus data. The map just explodes with information. Suddenly, you see the dense, tightly packed financial district of proliferative cancers glowing red. You see the winding suburbs of the supportive tumor cells in green, and crucially, you see the military base of T cells colored blue positioned far away from the tumor core, unable to penetrate. Once we have this colored in map, the real scientific detective work begins. We can start asking special questions. For example, we can run analysis to find out spatially variable genes. These are the genes that are switched on only in certain locations. We might find that the gene is active at the invasive front where the tumor meets the healthy tissue. This is a massive clue telling us that a gene is likely a key player at the process of invasion. This moves us beyond just cataloging cell types and into understanding cell states. The same cancer cell type might behave completely different and switch on a different set of genes simply based on whether it's in the core of the tumor or at the leading edge. The abstract map tells us who's in the city, the spatial projection tells us what they're doing there and why and where.
That's a very powerful distinction. So you can see not just cells, but their localized behavior. So, how do you take that one step further to understand how these different cell neighborhoods interact, and how do you decode the social network of the tumor?
This is where we get to the heart of the ecosystem concept. And it's driven by a suite of computational tools for neighborhood analysis and cell to cell communication inference. Since we know the precise coordinates of every single cell, we can computationally ask questions that were impossible before. We can, for every single T cell, count how many cancer cells are within a 50 micron radius. We can ask, are macrophages more likely to be found next to the vessels, blood vessels, or do certain types of fibroblasts form a physical barrier between immune cells and cancer cells? This is a digital pathology at an unprecedented scale. We can go even deeper by modeling communication. We have databases of unknown ligand receptor pairs, the proteins that cells use to talk to each other. Our algorithm can walk through the spatial map and look for neighborhoods where a cell is expressing a specific ligand and its adjacent neighbor is expressing the corresponding receptor. When we find these pairs, we can infer that the conversation is likely happening. By doing this across the entire tissue, we build a comprehensive map of the communication architecture. We might discover a hidden conversation where a small group of stromal cells is sending a go to sleep signal to nearby T cells, creating a localized immunosuppressive niche. This allows us to pinpoint the specific interactions that are driving the tumor success. We are essentially moving from a static census to a dynamic social network map, identifying the key conversations we need to intercept.
Right. So beyond the direct neighborhoods, the tumors are known for their larger architectural patterns like immune excluded zones or highly vascularized regions. So how do you computationally identify and define these larger districts or boroughs of the tumor city?
That's the fantastic questions because it's about seeing the forest, not just the trees. To do this, we use methods for spatial segmentation or tissue domaining. Instead of looking at individual cell neighbors, these algorithms look for larger regions with statistically similar characteristics. For example, an algorithm might find a large domain spanning thousands of cells. This is characterized by low immune cell density, high expression of genes related to hypoxia, low oxygen, and a specific subtype of cancer associated fibroblasts. We can then label this entire region as a hypoxic immunosuppressive domain. Think of it like a real estate developer uh, analyzing a city. They don't look at individual houses, they look for overarching patterns to define neighborhoods like the commercial district or the residential areas. Our algorithms do the same, but they use gene expression and the cell type composition as the data. By identifying this domain, we can simplify the complexity of a tumor into smaller number of functionally distinct territories. This is incredibly powerful because it allows us to correlate these large-scale architectural features with clinical outcomes. For instance, we might discover that patients whose tumors have a large hypoxic immunosuppressive domain are less likely to respond to immunotherapy, providing a powerful new biomarker.
This has been an incredible look under the hood of how to, decode complexity. But these are all inferences, re engineering the ecosystem to be hostile to cancer a sense. So that's a very powerful and hopeful note to end on. And if you could give one piece of advice to a young biologist starting in this field, what would it be exactly?
it would be a it would be to embrace the duality. The future of biology does not belong to the pure wet lab biologist or the pure computational biologist or the computational scientist. It belongs to the people who can act as a bridge between the two worlds, learn to code but never lose your intuition for the biology. Understand the statistics, but never forget the patient behind the data. The biggest discoveries in the a new era of spatial biology will be made by those who are fluent in both the languages. Because only by integrating the deep complexity of the data with the profound elegance of the biology, we can hope to solve a problem as complex as cancer. Be the translator, be the bridge.
Right. So, be the bridge, a very perfect piece of advice to conclude our series. From the architecture of cancer to the geography of its evolution, you've already given us a masterclass in this revolutionary field. Rudranshu Paul, thank you so much for joining us and sharing your incredible insights.
Thank you, ma'am. It was my pleasure. Thank you for this wonderful conversation.
It's a wonderful conclusion. To defeat cancer, we must understand that we are not fighting a single rogue cell, but an entire adaptive world. And to see that world, we need a new generation of map makers. This has been the Noisy Code. Thank you.